Hello students, welcome to Misostudy. So in the today's session, as we are dealing with the chapter that is Principle of Inheritance and Variation, we will be talking about a new topic, that is a linkage and recombination. So the topic is linkage and recombination. Linkage and recombination. Linkage and recombination. Till now we have studied that each gene or each allele, they segregate during gamete formation. Each gamete, they all are having alternating form of that gene. We have studied about the segregation of genes, right? In the Mendel experiment, we have thought that, and we have said that the chances of segregation of two alleles, chances of segregation of two genes, they are equal. For example, we were talking about one experiment. Let's take one experiment which is related to round and yellow color pea plant. This experiment I was doing, and in that experiment in F1 generation, what was the result? The result was this, right? This was the result in F1 generation. Now, in the F2 generation, what was the case? What were the different kind of gamete which were formed? So when the selfing was done, the gamete formation that comes out to be like this. Capital R can combine with a capital Y, capital R can combine with a small y, small r can combine with a capital Y, and the small r can combine with small y. These are the different type of gametes which can be formed by one of the parent of F1, which is a result of an F1 generation. So can you see that equal opportunities are there for R to combine with a big Y, and again R to combine with a small y. So equal opportunities were there. This we have studied in Mendel. Now, problem is that whenever the genes, they are located on same chromosome, the chances of segregation of them becomes very less. Lesser the distance between the two genes, more are the chances that the same type of genotype will be present in the daughter progenies. That means, suppose these two genes, this capital R and the capital Y, this one and this one, if these two genes, they are present at a very small distance in a chromosome, a very small distance, suppose they are present over here and here, right? Chances that during crossing over, these two genes will not get segregated. Segregation means this gene will be transferred to the next chromosome. So there is an equal opportunity for the transmission. But linkage said that this particular process says that if two genes, they are in closely contact with one another, lesser the distance between them, chances that they will be present like this in a daughter progenies. Whereas more the distance, the chances of segregation is more, right? So depending upon that, a very beautiful experiment was done by T.H. Morgan. He worked on Drosophila. Now the question arises, why we always use Drosophila? Why we use Drosophila? What is linkage? What is recombination? Then we'll proceed towards the experiment which was done by T.H. Morgan, right? So hope the things are clear to you that in which direction we will be going today. Linkage and recombination. So work was done by T.H. Morgan. T.H. Morgan. And he worked on Drosophila melanogaster. Drosophila melanogaster, right? This is is also called as a common name of a Drosophila melanogaster is fruit fly. Now, why always experiments are done with Drosophila melanogaster? The reason is that it is easy to breed these line in a laboratory condition. That means we can maintain them in synthetic medium. Easily maintained in synthetic medium. Easily maintained in synthetic medium, right? Which means they are easily cultured in laboratory. Right? Another reason is that when we try to culture these cells, they can produce large number of eggs. The female can produce a large number of eggs. Female can produce 100 of eggs at one time. Let's look at the life cycle. Drosophila melanogaster can complete its life cycle within 2 weeks. So, life cycle 2 weeks. Right? And they are easily available throughout the year. So, easily bred throughout the year. Throughout the year. Now, another reason is that the Drosophila melanogaster, we can easily see those variation with the help of a normal microscope or a low power microscope. Now, let's discuss that later on. First, let's talk about the genetic constitution. Overall, 4 different chromosomes, they are present in them. Drosophila has 4 morphological different chromosomes. 4 different morphological chromosomes they have, right? So, this is a Drosophila melanogaster. Now, along with these 4 different morphological different chromosomes, the variations can be seen, and these variations can be easily visible with the help of low power microscope. Variation seen by low power microscope. Low power microscope. Now, one more thing. When we talk about the XX and XY chromosome, that means a sex chromosome, male constitute X and Y, whereas a female has same as that of a normal human female, that is XX chromosome. So, male, they are having XY chromosome, and female, they are having XX chromosome. So, that is why study is being done with the help of Drosophila melanogaster. Now, we are studying about linkage and recombination. Linkage means how many, what is that frequency of formation of recombinants. That means it is that frequency by which the two genes or a pair of a gene, they are separated. Let's write about linkage. Linkage means if two genes, they are closely associated with each other. If two genes are closely associated with one another, chances of their segregation during gamete formation is very, very less. Very, very less. If two genes are closely associated with one another, chances of their segregation during gamete formation is very, very less. That means number of recombinants in this case will be less formed. So, more parental types, more parental type will be formed. More parental type will be formed, and one more important line, that means less recombinants will be formed. Less number of recombinants will be formed. Now, what we have learned because of this? More closely they are associated, the chances of separation is very difficult, and more distantly they are located, more number of the recombinants will be formed, right? So, this is called as a linkage. Let's write about recombination. More the distance between genes, more the distance between genes, chances of segregation is high. Chances of segregation during crossing over or because of crossing over is very high. Right? So, in this way we can calculate the whole recombination frequency. By calculating the number of recombinants upon total number of offspring which are formed, we will get to know about the recombination frequency. Recombination frequency gives us an idea that how apart these genes they are located. More the recombination frequency, that means more will be the distance between the genes. So, I have written over here, more the distance between the genes, chances of segregation because of the crossing over is very high, and more recombinant that ultimately means that the distance between genes are high. Right? That's why crossing over is easy. More distance they are located, easy for the crossing over to occur, easy the chances of segregation, the chances that in the gamete they will be segregated, more number of the recombinants will be formed, less number of the parental diatype will be formed. This is called as a recombination and this is called as a linkage. Now, the experiment which was done by Morgan As I have already discussed, this was done on the Drosophila. How this experiment was done, let's look at this. This experiment was done in two forms. One is yellow bodied and white eyed, which is a recessive form, whereas another, the same, it was crossed with a brown bodied and red eyed, right? So the experiment was done. So this was a Morgan's experiment on Drosophila. Morgan's experiment on Drosophila. As we have already discussed, the variations are there, and these variations, they can be seen with the help of a low power microscope. In that, he took two characters. One character was related to white body yellow bodied and white eye. Yellow bodied and white eye. Yellow bodied and white eye Drosophila with brown body and red eye. How this experiment proceeded. This yellow body and the white eye, he took it as a female. He took this female, and in this condition, they took a male, right? Yellow bodied and white eyed, they were recessive condition. The whole yellow body, they are what? Recessive condition. Whereas this a brown body and the yellow eyed is a dominant condition. Dominant condition. In this case, the experiment which was done, let's look at the whole experiment, let's look at the whole crossing over. So students, a very important experiment. So, the yellow bodied and white eyed. So, yellow bodied, that means that this is a yellow body and white eyed. So, let's draw chromosome structure only. Right. Another was a male. Right. This is a male, whereas this is female. In the female, the two type of genes they are present, that is yellow and the white eyed. Right. In this case, the condition was a brown bodied and red eyed. So, I am just writing this as a Y plus and another as W plus. Why I have written this towards this side? Because they are considered as they are linked. So, they were present only one side of, they were present around in one side of centromere. So, chances of segregation is very high. So, this is what? This is a female. Whereas this is what? This is a male. Right? Now let's discuss about what type of generation will be formed in F1. In case of F1 generation, In case of F1 generation, what will be the case? This will come towards this side like this, and they will be having a yellow like this, The morphological feature between the male Drosophila and the female Drosophila is that the male Drosophila is a smaller one, whereas the female Drosophila is a bigger one. Now, in this case, what has happened now? Again, the Y and the W, that is the yellow and the white-eyed, is present like this only, whereas exchange has taken among these. Now, these are Y plus and W plus. Again, these are Y and W. Now tell me one thing, what will be the phenotype of these? The phenotype of these will be a brown bodied and red eyed female. Brown bodied and yellow eyed, red eyed female. Right? So opposite occurred. In this case, what will be the scenario? Yellow bodied and white eyed male. Right? This is our F1 generation. Now there is a formation of F2 generation. After this F2 generation, the cross has done between these F1. After the crossing of F1, what we got is that the parental ditype, they were around 97%, and the recombinants, they were around 1 point something, right, around 98.3, I guess, 98.7 we had this parental, whereas 1.7% we had only recombinants. So after that, the crossing has done in these type of individuals in the F1. So can I write over here intercrossing in F1? Intercrossing in F1 generation. Intercrossing in F1 generation. Now what will happen? Let's talk about first about male. Obviously in male, the Y chromosome, they will be present like this. 4 different type of offspring will be formed. Right? In one case, the chances that yellow and white-eyed will be there, the male will be there. Another case is that, so I need a red color for that. So hope you can distinguish. So what will be these? These will be a brown and red-eyed. In this case, number of the recombinants formation will occur. Let's see. In some cases, one yellow will come like this, and the chances that this W plus will come like this. There is an insertion, so this is a recombinant formation, right? Or the chances are that there is formation of a yellow plus, or chances that the next all will remain same. So there will be a normal W, right? So these are what? These are like that of parents. These are parental, and what are these? These are called as a recombinants. So can you tell about the phenotype of this? Obviously the yellow bodied in the white eye male. In this case we have is another, that is a brown bodied and red eyed. In this case the yellow bodied and red eyed male. In this case the brown bodied and white eyed male. So such type of recombinants will be formed in crossing over. So let's write about these. What are these? Yellow bodied and white eyed. White eye. These are this. What about these? These will be a brown bodied plus this red eye. All these are males, right? What about these? These will be yellow bodied and this will be red-eyed. So what are these? These are recombinants. And what about these? These also will be brown-bodied, brown-bodied and white-eyed male. So this will be the phenotype of offspring. Now, let's talk about males. How this males, let's talk about females. What will be the genotype of females? Let's see. The another. So, this is a female. Chances that they may get such type of phenotype. Now, let's write about the phenotype. The chances that there may be a YW, that this will also be a YW. In this case, that this is Y plus and a W plus. In this case, there will be a Y and a W. In this case, what has happened? The changes have taken place. The chances that in this case, the Y plus W formation has occurred, right? So again, we have, so the chances that this is Y and this is W plus, again this is Y and W. So these are the different 4 type of female. Out of these are parental and these are what? Recombinants, because can you see the changes has occurred. That means it shows that the crossing over have taken place. So the crossing over have taken place, that means the number of recombinants, they are formed a very, very less because the distance between them is very less. So what he found, that these yellow and the what will be the phenotype of these? This will be a yellow bodied and white eyed. What will be the phenotype of these? I know that these are dominant, so that's why they will be a brown bodied and brown bodied and red eye. This will be again a female. In this case, what will be the scenario? This will be a brown bodied and white-eyed. In this case, this will be a yellow-bodied and red-eye. So, on one side, I have written about these are parental types. On this side, I have written about the recombinants. When we talk about the whole recombination frequency, the recombination frequency was lesser. So the distance between the gene was lesser. So the parental types, they were more, right? So come at this side, students. We have done about the Morgan's experiment. In that Morgan experiment, they were the parental type. Right? Now, when we talk about the whole frequency, so that was around 98.3% are parental, 98.7% it was there, they were parental, and around 1.3% there is a formation of recombinant. they were recombinant, right? Hope the whole experiment which is related to the recombinant, this was first experiment which was done on Drosophila. Simultaneously another experiment is also done, that I will discuss in the next class. If these things are clear to you, side by side write all these diagrams in your notebooks as well, because at this time you might be feeling okay, the things are understood to you, but you will be able to do the same only if you draw the same in your copies. So students, will meet in the next lecture. Till then, take care and thank you so much students for watching this.